Imagine if your immune system carried a personalized barcode, constantly scanning every cell to decide friend or foe. That barcode is encoded by the human leukocyte antigen, HLA system, a genetic masterpiece that governs immune surveillance and self-recognition. The ability of the immune system to distinguish self from non-self relies heavily on a remarkable genomic region known as the major histocompatibility complex, MHC, or the human leukocyte antigen, HLA system in humans. This system is located on the short arm of chromosome 6 and encodes cell surface molecules essential for antigen presentation to T cells, a cornerstone of adaptive immunity. MHC molecules responsible for antigen presentation fall into two primary classes. Class 1 MHC molecules are expressed on the surface of all nucleated cells. These transmembrane glycoproteins consist of a polymorphic alpha, heavy chain paired non-covalently with a conserved B2, microglobulin. The heavy chain comprises two peptide binding domains, A1 and A2, an immunoglobulin, Ig-like domain A3, a transmembrane domain, and a cytoplasmic tail. Genes encoding the class I heavy chains are found at the HLA-A, HLA-B, and HLA-C loci. CD8 plus cytotoxic T lymphocytes, CTLs, recognize antigens presented by class I MHC molecules. This interaction is crucial for detecting and eliminating virus-infected cells or malignant cells. CD8 binds to the conserved region of the MHC class I heavy chain. In addition to classical class I molecules, non-classical class I molecules such as HLA-E and HLA-G play specialized roles. HLA-E interacts with NK cell receptors, while HLA-G contributes to maternal fetal immune tolerance. Class II MHC molecules are primarily expressed on professional antigen-presenting cells, APCs, including dendritic cells, macrophages, B cells, and thymic epithelial cells. Expression can also be induced in other cells by interferon gamma, IFNG. These molecules consist of two transmembrane polypeptide chains, alpha and beta, each contributing to the peptide binding cleft. Class II MHC genes reside in the HLA-DP, HLA-DQ, and HLA-DR regions of chromosome 6. Antigen presentation by class DQ molecules is recognized by CD4 plus helper T cells, which coordinate immune responses through cytokine secretion and B cell activation. While not involved in antigen presentation, the MHC class III region encodes several immunologically important proteins. These include components of the complement system, C2, C4, and factor B, pro-inflammatory cytokines like TNF-A and lymphotoxin, and heat shock proteins. HLA alleles are identified by a standardized nomenclature, the gene name followed by an asterisk, then a numerical designation, for example A02, 01. The first field indicates the allele group, often correlating with serological specificity. MHC molecules are among the most immunogenic antigens in the context of allogeneic transplantation. The strongest determinant in graft rejection is HLA-DR, followed by HLA-B and HLA-A. Accordingly, these loci are prioritized during donor-recipient HLA matching. Certain HLA alleles are also linked to susceptibility to autoimmune diseases. HLA-B27 with ankylosing spondylitis and reactive arthritis, HLA-DR2, DRB1602 with narcolepsy, HLA-C06, 02 is linked to psoriasis, HLA-DR4 to rheumatoid arthritis, and HLA-DQ2/DQ8 to type 1 diabetes mellitus. Understanding the HLA system offers critical insights into immune recognition, transplant compatibility, and the genetic basis of immune-mediated diseases. Subscribe and follow for more in-depth explorations of how your body defends itself at the molecular level.